Hey class, it's Mr. Durst here, and in this recording we're going to focus on exercise 12, microanatomy of the muscle. In your lab manual, this is exercise number 12, and you can reference figures 12.2 and 12.5. Invisible body in the A&P app, it is chapter 14, section 6, or chapter 14, section 3. I also want to emphasize that we have some microanatomy models as well. So I'm going to go check out Visible Body. I'm here in Visible Body's Anatomy Atlas. And instead of the systems view, which is the default view, I'm going to scroll over here, not to cross sections, but to microanatomy. And from microanatomy, I'm going to scroll down to muscular system and click on skeletal muscles. So here we have the microanatomy view. And we're just going to go down the line and highlight some of these things that we need you to know. First, we want you to be able to recognize the muscle fiber, or the myocyte. So an individual muscle fiber is going to be right here. And the cell membrane of the muscle fiber, is right here, the sarcolemma. So this is one individual muscle fiber that I have highlighted. And because only the outer membrane is selected with Invisible Body, it's being called the sarcolemma. So the muscle fiber or muscle cell known as myocyte is this entire grouping of bundles that I have on my screen right now. If I go to multi-select, I can go bam and just select a whole bunch of them all at once so you can get a better idea of what we're looking for. So this whole thing is a muscle cell, a myocyte, or a muscle fiber. The cell membrane of a muscle cell, or muscle fiber, is called the sarcolemma, and that's what I have highlighted right here for us. And as we're working our way down, within the muscle cell, we have these large bundles of small, tiny filaments. These bundles are referred to as, let me highlight them all there we go, the myofibrils. So it's the whole bundle. And if we take a look at this muscle cell membrane, I want to emphasize here that we have some holes in the cell membrane. These invaginations or holes in the muscle cell membrane correspond with a network of tubes that goes through all the way through the entire muscle fiber out to the other end. This network of tubes that goes all the way through, that transverses the muscle cell, is referred to as the T-tubules. And if we look at these T-tubules, which is right here on our lab objective list, the T-tubule is going to be right next to the sarcoplasmic reticulum. And in particular, if we zoom in here, I'm going to go ahead and annotate the screen. Let's go with my favorite color, green. We can see how there's two bulges of the sarcoplasmic reticulum right next to an individual T-tubule in between them. This group of three is referred to as the triad, one T-tubule and two terminal cisternae of the sarcoplasmic reticulum. As we are looking at the muscle cell, it's also important to emphasize, or muscle fiber, the myocyte or muscle fiber, that they need a lot of mitochondria, a lot of ATP, so they're going to have a lot of mitochondria to make those ATP. And these mitochondria are shown in yellow, kind of goldenrod here in Visible Body. We also, within a myofibril, can take that myofibril and break it up into segments. So if I look at one myofibril right here, here's a section of here's a myofibril that has had its sarcoplasmic reticulum and T-tubules removed, and you can see how individual segments of the myofibril could be highlighted one segment at a time. This one segment of a myofibril is referred to as the sarcomere. And as we look at the sarcomere, you can see there's this zigzaggy line that marks the end of one sarcomere and the beginning of another sarcomere. So I'll go ahead and just scribble on that zigzag for us here, since VB is not letting me highlight it. So that zigzag right there, that's the Z disc, separating one sarcomere from the adjacent sarcomere within a myofibril, or within a muscle cell, within the muscle cell. And then in the very middle of the sarcomere, we have the M line. So the Z disc marks the beginning and ending. The M line marks the middle of the sarcomere. So if I zoom out here, you can see there's this 1, 2, 3 bands right in the middle, or 3 lines right in the middle of the sarcomere. That middlemost line right there, right in the middle, that's the M line, which stands for middle. And when the sarcomeres contract, the thin and thick filaments that make up the sarcomere will overlap by an increasing amount. So we can see the thin and thick filaments shown as, let me clear the screen here. Here in yellow, we have a filament, and then here kind of in a darker color, we have another filament. They're horizontal on the screen and parallel to each other. These thin and thick filaments are going to be pulled towards the M line, or pulled so that they overlap more during muscle contraction, when the sarcomere is contracting. So we are not going to ask you to differentiate between thin and thick filaments. Just know that collectively these are myofilaments. We have the muscle fiber. Inside of the muscle fiber, we have myofibrils. Inside of the myofibrils, we have myofilaments. Some of them are thick, some of them are thin myofilaments. So when you get a chance, please boot up Visible Body class and spend some time playing around with this. It is a fantastic microanatomy teaching model. That's all we have for this recording. If you have any questions or comments, please feel free to post them in the class discussion board. And as always, happy studies.